Hello, my name is Jeffrey Han. In this video, I will be describing a general method to treat autoimmune disorders that are mediated by T cells, particularly a disease called aplastic anemia. In our body, T cells are an important immune cell type that protect us from infection from pathogens. However, in certain individuals, a small minority of T cells attack our own body, resulting in a wide array of diseases termed autoimmune disorders. In aplastic anemia, a very small population of T cells target our bone marrow cells, which impairs our ability to create new red blood cells as well as cells of our immune system. The vast majority of T cells, however, do not attack our bone marrow. The current treatment for aplastic anemia involves treating patients with antibodies that recognize and kill virtually all T cells, both good and bad. A more ideal treatment would be to eliminate the bad T cells while not affecting the good ones. As an analogy, let us consider these rocks. On the left are good rocks who live freely, while on the right are bad rocks who are in prison. Let's say one of the good rocks committed a crime. If we consider my hand to be law enforcement, it would be unreasonable to imprison all the good rocks just to guarantee we catch the bad one. Since the rocks differ from one another in characteristics like shape and color, we can target certain rocks that are bad. Similarly, T cells differ from one another by virtue of their T cell receptors, or TCRs. Different TCRs recognize different proteins. Good T cells have good TCRs that recognize pathogenic proteins and can kill pathogens, while bad T cells have bad TCRs that recognize our own proteins and attack our own cells. Using modern immunological and genetic techniques, we can determine which TCRs are responsible for T cells killing our own cells. With respect to aplastic anemia, we first determine the proteins that bone marrow cells make, but T cells do not make. Since bad T cells exist, they likely aren't making the bone marrow protein that their TCRs recognize, or they would just end up killing each other. Next, with experimentation and bioinformatics tools, we can determine which region of the bone marrow specific protein bad TCRs recognize. We can then purify the bad T cells using this particular region of the bone marrow specific protein. Using modern cloning approaches, we can identify and clone the TCRs that this bad T cell makes. Next, we can then produce antibodies against just these bad TCRs. Finally, we can use these antibodies to treat patients with aplastic anemia. Since these antibodies only target bad T cells, the good T cells will be unaffected. This contrasts from the current treatment of aplastic anemia, where patients are given antibodies that target and kill all T cells, both good and bad. Using the approach described, we can treat autoimmune disorders like aplastic anemia by eliminating only those T cells that are bad, leaving the good T cells to protect us from pathogens.